All right. Good evening to all of our guests, parents, Board of Ed, council, different commissions here, the Tree Commission, and other VEC members. This evening marks a culmination of an entire year that our two junior commissioners, CC Manis and Nia Polari, have been working with us. The Junior Commission program was established last year to engage and work with Verona High School students who exhibit an acute awareness of and an interest in our environment and our ecosystems and the problems that we face to maintain a sustainable future. These two young women have both demonstrated a deep dedication to work both independently and as our teammates to investigate and address some of the issues that are local in our community and the world beyond. As you'll soon hear, both Nia and Cece have stayed very true to their primary interests over this entire year by doing deep dive investigations into those topics that most interest and inspire them. And after each student's presentation, there'll be time for any questions that any of you might have. First, I'd like to introduce Cece Manis. Cece is currently a sophomore at VHS and came to us as a freshman last year. She brings energy and a wonderful sense of enthusiasm to the VEC, whether she's at meetings with us or at our events. In school, she's a member of VHS's varsity swim and cheer teams and swims for the Cougar Aquatics team. She's an officer in the VHS Mandarin Club and does social media and writes articles for the school newspaper, and she volunteers at the middle school. Out of school, she enjoys hanging out with her friends and working with the Great Swamp Association to test Verona's waterways. I do not know when she has time to do that, but she does. The VEC was first introduced to Cece through her application a full year ago, last December. In response to one of the questions, Cece wrote, "My current biggest concern is Verona's waterways. I've taken a big interest in water pollution and have a deep understanding of the history behind America's Water Act. I wrote an essay for the contest National History Day about the Clean Water Act of 1972. I know which ideas worked and which ones failed. Water pollution scares me because humans rely on water for many reasons, and the thought about what could be floating and living in the water because of point and non-point source solutions scares me." Without further ado, here is Cece on water quality in Verona.
Hello, everyone. I'm CC Manis, and I'm so excited to be here tonight to talk to you about my research on Verona's water quality. So I just want to give a brief background as to why I chose this for my junior commissioner topic. Jessica already addressed it a bit, but I first found interest in waterways in 8th grade, a class I took at HBW Middle School National History Day. Here, I wrote an essay about the Clean Water Act in 1972. I was able to make it past the regional and state levels of the competition all the way to the national competition, where I managed to place 7th in the nation in my category. Since then, I got trained with my mom and my brother by the Great Swamp Water Association of New Jersey to test New Jersey's waterways, and you'll see that my training has implemented my research today. So first, I just want to talk about the current state of Verona's water. So the Township of Verona has been working on removing PFAS from the Lynn Drive and Fairview Avenue wells. Water bills for Verona citizens are rising, and Verona currently purchases their drinking water from the Passaic Valley Water Association because our water is not sanitary enough to drink. So what else can be highlighted? Well, I addressed this in my research through the Peckman River. So first, a brief background on the Peckman River. The Peckman River is a tributary of the Passaic River, which means it is a small water stream that flows into the larger Passaic River water stream. Some believe the name of the Peckman is to be named after the Lenape word for cranberry, which is pecan, but it is most likely the surname of an English settler. It runs between the first and second Watchung Mountains, and its source is located in the Eagle Rock Reservation, which is in West Orange. So West Orange, the river starts in West Orange. It runs 8.5 miles, and it converges with the Passaic in Woodland Park. And two notable locations along this river are the Devil's Hole in Cedar Grove, and what we all know very well, the Verona Lake in Verona. So basically, the Devil's Hole used to power a steel factory back in the day, but since then the steel factory is shut down, but there still remains near the hole today. And then the Verona Lake, which used to power a grist factory, it got dammed out, the swampland, and now created the lake we know as the center of Verona Park today. And then in this graphic, you can just kind of see the area inside the cliff shows where the Peckman River flows through Verona. Later in my presentation, I focus on how our state of the Peckman affects the creatures found in here. So first, I'd just like to build some emotional connection and look at the stats from an NJGov report on what fish are most found in the Peckman River. Here we see the creek chub, black-nosed dace, brown bullhead, goldfish are all very common fish found in the Peckman. And obviously, as an environmental commissioner, junior environmental commissioner, I had to just quickly address the Verona Environmental Commission's history with the Peckman. So the Verona Environmental Commission has held cleanup events near the Peckman. They've surveyed land around the Peckman River that notes the loss of riverbanks. And they had also drafted and sent up cleanup recommendations to the town council. And the town council was then to pass these on to the Essex County office, as Verona cannot directly make any laws according to the Peckman, since it is like a county property. Also, through sitting in on the Verona Environmental Commission meetings, I see how they work to honor the Verona Stormwater Ordinance. Every time new major and minor infrastructures are being added to Verona, they make sure that it meets all the impervious surfaces, runoff and impervious surfaces. So going into more specific niche location of my research, I sampled between Park Place and Derwent Avenue, where the Peckman runs between those two streets. The river flows below the ground level, making it very accessible to runoff. But it is important to note that there was no rainfall the week preceding my sample collection, so I did not really have to take into account runoff in my research. So what were the key data points I was looking for? Well, I was looking for the pH, phosphate levels, and nitrate levels of the water. Just to better understand these points, I'm just going to give a brief introduction to each of them. So the pH basically measures how acidic or basic a body of water is. The scale ranges from 1 to 14, 1 being the most acidic water can be, 14 being the most basic, 7 being sort of like a neutral point. And in streams, aquatic animals prefer a pH range of 6.5 pH and 8 pH. And the pH might be affected by the environment which the water is running through, so how the current pollution is in the area or the soil surrounding the lake. Phosphate. Phosphorus is a nutrient used for plant growth, and it is found in low levels in the environment, but too much of it can lead to over-fertilization of the land. It is found in fertilizers, cleaning detergents, and soaps. And back to over-fertilization, high phosphorus levels can lead to eutrophication. This is basically when it leads to rapid growth of plants such as algae, and algae sucks up all the oxygen, it dies in the water, and this creates a problem for the aquatic creatures living in the water because they don't have anything to breathe, so they eventually start to die out. The ideal phosphate levels in rivers should be between 0.08 and to 0.10 ppm, which is parts per million. Nitrate, along with phosphate, is a nutrient that encourages plant growth. It is found in fertilizers, animal and human waste, and in decomposing organic matter. It also helps lead to the eutrophication of waterways, and that ideal nitrogen reading is less than 1 ppb. And here in the graphic, you can just kind of see the process where the runoff goes into the water, it creates algae, the algae dies, the fish suffocate. So what did I do to test for these? Well, first, I collected 3 test tubes, which were each filled with 10 milliliters of river water. A chemical tablet was placed into each of them to the respective data I was trying to find. And after 5 minutes, a color appeared that reflected the river's pH, phosphate, and nitrate levels. Then compared them to these sheets from the Great Swamp Watershed Association, and they gave me the results. So starting off with pH. Here you can see by its greenish color, the water had a pH of 7. This is a very positive result, as the ideal pH was between 6.5 and 8 pH. As for phosphate, it was not as positive. Here we can see by the light blue color that the phosphate of the water was around 1 ppm, when the ideal range is 0.08 to 0.10 ppm. This voiced a point of concern in my head. Along with that, the nitrate, we can see by the orangish color that the Peckman River had around 2 ppm of nitrate, when it should really have less than 1 ppm. So what can we conclude for these results? Well, while the pH of the water is optimal, the phosphate and nitrate levels are dangerous to our town. They can lead to the increase of algae blooms and a decrease of organisms in our water. So what does this really mean? What challenges does a river face without organisms living in it? Well, one, the loss of water quality regulation. Aquatic organisms, they filter out pollutants and organic material found in the water. Without these organisms, like such as fungi and bacteria, we just we don't the regulation is not being as circled out, and our water quality only begins to worsen. Second, sediment accumulation and changes in flow. As the sediment gets to build up, not only does it affect the current state and quality of our water, but it affects physically how our water looks, and it changes the flow, and it might block up certain areas of the river where water needs to get through. Lastly, the loss of biodiversity interactions. So rivers support ecosystems. They provide water, nutrients, and food for all the surrounding organisms. So fish out of the water, or birds out of the water, might eat the fish inside the water. Certain organisms might drink the water, and when it's not great quality, they might die from it. And this just ruins the entire food web of our environment. So, how can we solve this? Clearly, it is a problem. First, for the town, we could reduce the use of fertilizer. This could be done through ordinances that might say use organic fertilizers instead of synthetic fertilizers. While organic fertilizers might pose their own issues in their respective manners, synthetic fertilizers have very high levels of nitrate and phosphate, and we can't afford to have this increase. Lastly, we could also introduce an ordinance that encourages a limit on the amount of fertilizer consumed. I already know that the town does have some sort of a limit they use on the amount on their fertilizer to ensure that we're not adding to the runoff, or limit it as much as possible. But maybe we can start focusing on per citizen or per neighborhood or per lawn. Next, to monitor the recycling of soaps and detergents. Everyone thinks of fertilizer when they think of phosphate and nitrate, but really things such as such as detergent and soaps, also have these high levels of phosphate and nitrate. So maybe set up like a program or flyers around the town that encourages residents to not just leave their soaps and detergents sitting out on the street, but rinsing the bottles out and recycling them responsibly at Ozone Avenue. Lastly, we could plant buffers along the Verona Lake and the Pefin River. These buffers will prevent fertilizer runoff from reaching the river, because when it rains, they go into the dirt and the soil and the plants, and they're not going straight into our river. And it also introduces more green life to the environment. Well, let's focus away from the town and just what can you do as a citizen of Verona? Well, one, research fertilizers. Try to invest in organic fertilizers instead of synthetic fertilizers. Like I previously said, synthetic fertilizers just have very high levels of phosphate and nitrate that are only adding to the results I found. And some organic ideas are compost, seaweed fertilizer, fish emulsion. And you have to make sure to research your organic fertilizers because they're not all perfect. But if you just put that extra effort, it can really make a difference in the numbers. Next, make sure the trash ends up in the trash can. Soaps, detergents, and fertilizers have high levels of phosphate and nitrate, and if they are not disposed of properly, they can end up leaking their toxins into the river. Lastly, educate others. Verona is a very close and tight-knit community, and everybody wants to help out the state of our town at the end of the day. But our waterways are only going to improve if a majority of Verona citizens are willing to understand and take action. Do something about the fertilizers. Make sure they're rinsing out their bottles and disposing of them responsibly. So I'd just like to give a couple of thanks, one to the Verona schools and staff for providing my classes and great teachers and just encouraging learning. Then the Verona Township, I definitely got to dig into more of what the township does by looking at their meeting minutes and their past ordinances. And I really got to get a deeper understanding of what our town's doing for us and realizing that it doesn't all come naturally. The Great Swamp Watershed Association, I got all my research materials from them, and I got my license almost to test the waters in New Jersey. So couldn't have done it without their help. And lastly, the Environmental Commission for giving me this opportunity, for welcoming me into your community and giving me improvement, letting me go to park cleanups as pictured on the right, and giving me this experience to present in front of you all. Thank you.
Does anybody have any questions that they'd like to ask?
Yes. I like the thing about the point about planting a buffer around Verona Lake, because that would also help with stopping the geese from wanting to come to Verona Lake, and their feces contribute to this as well. So that was a really good point. That was my favorite part of the presentation. Thank you.
Cece, you said, did you test at the one spot just before the river went under the Hill Avenue?
Yes, you're right. It was between Derwent and Park Place, and it's like kind of where it flows underneath Bloomfield into the park.
Oh.
So if that paints a picture, almost it's between like the
So you were on the
Yeah.
on the far.
if you're walking Bloomfield and then you're on the side closest to Evan Brown, it's like that little gap where you look over the river and you see between the buildings.
So the runoff from there came. You're mostly coming from the golf course and the park, but then the other arm of is coming down from inside the neighborhoods of Verona too.
Correct, definitely.
Tributary. Okay.
Correct, yes, and it's like below the ground level, so it's like if you were, we had to kind of climb back there to get to the testing area.
Yeah.
So you can see even from these buildings, like who knows what's really running off from like the back pipes down to the river. It's very accessible. It's
Right.
almost equivalent to point source pollution in a sense, which is just basically when the pipe flows directly into the river instead of indirectly going there.
Like.
Oh, sorry, did you have a follow-up question? Okay.
Why did you want the president?
Oh, yes. Well, Jessica briefly mentioned that, more of talking about credentials and everything, but I wanted to join the environmental committee because I wanted to learn more about what was going on behind the scenes in our town. I mean, you can only learn so much from school. I mean, the clubs are great and everything, but it's getting to immerse yourself in like an actual committee and seeing the action they do. It's great. Even seeing what your dad does, he edits all our newspapers. It's really great to be behind the scenes and just learn more about our environment. And clearly I would not have done this research if it weren't for this. And I wanted to build off of what I had done in national history. I didn't want to end there. And I wanted to use my license through the Great Swamp Watershed Association for good for our town.
Quick, would you recommend testing at the source of Verona Lake from what comes off the golf course and then comparing that to what the end of the lake or where you took your site and then have different state? Would you recommend that and then go to the country club and ask them to maybe try to work on their runoff?
Yeah, well, for sure. I could definitely do that. I mean, I have all the materials at my house. I could get back to you on that follow-up presentation next year, guys. I can bring the gang back. I can do a little comparison if you'd like. But yeah, no, I could definitely send you some results. I mean, we were going to go testing, my mom and my brother and I actually, and definitely do the lake. I think that's a great idea too.
When we were down there was like a little sludge on top of the water. Would that be coming from the lake, or would it be coming from like the buildings on the side?
Like, what do you mean by that
So,
specifically?
like there was like a sludge on top of the water, kind of.
Is it like
that? Yeah, like a film that was.
Well, that could just be point source pollution, like the runoff actually coming from the buildings. Or it could be, like I said in the presentation, nitrate and phosphate, like it does cause sediment buildup, and maybe that could be early signs of sediment buildup. So much could be hypothesized.
Any other questions? All right.
Jeff, I just have a comment real quick.
Sure.
Cece, it was wonderful. I learned so much just by your presentation, so thank you.
Thank you.
And just hearing you speak, I can't wait to see where you go in life, because very impressive.
Thank you. Thank you so much. Thank you for coming out.
That was amazing.
Thank you.
It was really well done. And the slide deck was gorgeous. It really was. So, our next and final, uh-oh. I did something. So you'll get it back to where it needs to be as soon as you get up here. Is Nia Polari. who came to us as a seasoned sophomore last year and is currently a junior. Nia, a pensive and methodical thinker, is driven by data and data analysis. Currently, Nia participates in the VHS track and tennis teams. She's the chief editor of the Avant-Garde Club and vice president of Model United Nations. She also volunteers at the American Museum of Natural History. An amazing fine artist, Nia loves to draw and paint and actually has a history with the VEC, as she won one of the poster contests when she was at HBW many moons ago. And incidentally, the artwork that you will see throughout Nia's slides is also all original works of her art. Even out of school, Nia has always been an environmentalist, observing plants, animals, insects, and people, noting their similarities and differences and wondering what connects us all. She's interested in the sciences, especially microbiology, but she's also hungry to learn anything and everything throughout the entire world. Included on Nia's application from a full year ago, the following interests were listed, or were included rather. As a junior commissioner, I would like to advocate for the implementation of an inexpensive way to both treat and test the soil of parks and other reservation areas through nitrogen concentration testers, combined with other forms of technology. Sure. The eradication of invasive species and the implementation of native plants and insects to balance the ecosystem in the area was one of her listed goals. So without further ado, I would like you to help me welcome Nia Polari for her presentation on microorganisms and their effect on invasive versus native species.
Well, let me get this up to the starting slide. All right. So I just want to say a big thank you to everybody for coming here, and just a big thank you to the Verona Environmental Commission, especially for just giving me this opportunity to research and effectively put together this presentation to be shared with everybody else, because I think the topic that both me and Cece are going to present and have presented is just so essential for general knowledge and just understanding how life works. So my topic is how the microbiome and how microorganisms can be harnessed to act as biocontrollers against invasive species. And I think we all understand that invasive species have been on our minds as of late and have been for quite some time, and just finding solutions to eradicate invasive species in the area is just crucial to understand. Okay, so let's begin. So what I want to touch upon now is the microbiome. So what exactly is it? Now, we understand that it is a collection of microorganisms in an environment. And the microbiome is crucial for a variety of life functions. So that can range from both assisting with digestion in both humans, in butterflies, for example, on the surface of the plant, to the aquatic organisms in the ocean, because how will the aquatic organisms get their oxygen? The microorganisms do all that. The fungi are also included in with the microbiome, along with the right hand corner of that image, which is also a series of microorganisms that I found in my research. So an extension on how the microbiome assists with life is that it mediates balance. So understanding that microorganisms can balance pH levels in certain parts of the world or in the environment, both in the soil, in plants, in organisms alike, they're just crucial in doing so. They regulate chemicals, they regulate how chemicals go from one area to another, how it circulates throughout the environment. Recently, there has been a lot of talk about the gut microbiome and how it assists with brain function and other sorts of functionality within the body. And we understand that digestion is very crucial for human life and other life as well. So that has been a lot in the medical area too. And environmental supplementation. So nitrogen, and as Cece has stated in her presentation, phosphorus, phosphates, nitrates, all sorts of other nutrients found in the environment are just critical for sustaining growth within the environment and organisms within. So how do microorganisms exactly benefit plant species alike? So they're integral to providing nutritional supplementation, which does include nitrogen nitrates. It's critical for the mediation of soil. So mediate soil, what do I mean by that? Understanding how it balances out pH, nitrate levels, all sorts of levels within chemical levels, so proper plant growth can be sustained within the environment. pH regulation, as previously stated, and also it provides balance both chemically from outside and internally in the plant. And within this image right here, it explains how there's a variety of spheres, as in microorganisms are both within the soil, within the roots of the plants, on the surface of the plants, in the air. They're practically everywhere, mediating everything that occurs. Without microorganisms, we need to understand that plant life cannot function, normal life cannot function. Microorganisms were there to begin with and have assisted as a result with mediating growth and sustainability in the environment. And this can be also referred to as microbial symbiosis, which will be essential as we go deeper into this presentation. So now we need to understand invasive plants and how do they exactly negatively disrupt native plants. Now we need to understand also that invasive plants are plants themselves, so they also benefit from microorganisms. However, invasive plants in this respect are damaging to both the native plants and the exotic plants that are within our environment. Exotic plants being those that aren't necessarily affecting the native environment, but are rather just existing. So some statistics that I have found from the United Nations Agricultural Commission is that the U.S. Forest Service from the United Nations Commission estimated that about 42% of the native endangered and threatened species are affected by invasive plant species alone, 18% of which are currently being directly affected by the native species. And according to the Hilltop Conservancy, which I found on their website, more than 33 invasive plant species populate the reservation. Now that is a lot. It's a lot of invasive species that are now currently riddled around the Hilltop Conservancy, around the local area, and just affecting a bunch of the natural environment that we need to sustain the natural ecosystem with. So how do they exactly affect the microbiome and the native plants around? They can release chemicals within the soil, which can affect the microorganisms and the diversity. Now, when it affects the diversity, generally speaking, the more diverse the microbiome is within the soil, the better chances that the plants in the area can sustain themselves and grow. And microorganisms do exactly the opposite of that. They take out the biodiversity. They kill off microorganisms that are essential for these native plants to sustain themselves. And by doing so, they can also harness these microorganisms or introduce new ones so then they can extract nutrients, which we will go into as nitrogen deposition and extracting nutrients from native species in the soil, which is another aspect which is absolutely crucial in understanding. They can also disrupt the diverse biomass, as I recently discussed, in the biodiversity of the soil, along with the microbiome material within. And one aspect that I really want to discuss with everyone is that there have been efforts where invasive species have been taken out physically, and that has done a lot of work. That has done a lot of assistance to eradicating the local invasive species in the area. However, given that they can affect chemically both within the soil and the native plants in the area as well, in their vicinity, they can leave these same chemicals when they are disrupted, and they can even greater negatively affect the surrounding native plants in the area and just cause more death and destruction along the way. So in this snippet of the presentation, I'm going to include some experimental analysis, which I did. Just to preface this, I have researched a lot with other studies that I used to then create these experiments, which involved a lot of preliminary studies involving reviews on garlic mustard, how they extract nitrogen, and the Japanese knotweed, and those two invasive plants in the area are going to be included in this presentation as my test sets. So this is kind of a diagram that I drew out where I initially took some samples from the Lenape Trail. It was kind of near the Prisoner Pond in the area, which is above Freedom Field. And I identified both the garlic mustard, which is in the center image on the left. That is the garlic mustard. It's an invasive species. And I identified the other invasive species, which I took in the same area, which is referred to as the Japanese knotweed on the far right next to the microscope. And in the center of it all is what we call the white snakeroot. And the white snakeroot is a native species in the area, which is gravely affected by both these invasive plants in the area. So I took both a sample of the native snakeroot, a sample of the garlic mustard, which is invasive, a sample of the Japanese knotweed, and a soil sample from a native control, which is the white snakeroot, which I put into a well-fertilized, well-sustained soil, which had no connection or it had no effects from the invasive species that were in the area. It was just its own type of soil, and I put the white snakeroot in there for about a week, let it marinate in the soil, and I got those exact samples from there. So the first test that I did was the nitrate quick test, which determined the concentration of the nitrogen in the soil. And along with that, as a second test, I did a microscopic analysis of the roots of these plants, just to identify, oh, what type of microorganisms in there. Now, I can't exactly say how diverse each plant has in their how much diversity there is within each soil of the microorganisms, because that involves a lot of data collecting, which cannot be seen or deduced from observing, but I focused on the rhizosphere, which is that highlighted area, the red, which consists of bacteria that are responsible for providing the plants in the roots proper nutrition. So the nitrate quick test. In this image alone, we see the scientific names of both the garlic mustard, the white snakeroot, and the Japanese knotweed. For determining the concentration, I took each of the samples that I collected. The first test tube in the center, as we can see on the left, is the native plant that was within the vicinity of both the invasive plants. And the one on the right with the white snakeroot is the same one, except it is our control. So it was in a normal setting without being in the vicinity of other invasive plants. And we see how on the top right, we see which coloring is for low nitrogen content and high nitrogen content. And the nitrate in both the Japanese knotweed and the garlic mustard is extremely high. And as for the white snakeroot, it is extremely low for both the test tube that was in the vicinity of the two invasive plants, but not as low as it was in the control. As for the microscopic evaluation from each root, we find here is a little playthrough on what exactly we are observing when we are going to be observing these microscopic slides. So we can see here that the little squiggly lines in the circles, well, microscope slide, are the bacteria or other microorganisms. When you see the dark little material there, that is the organic matter. Anything that is remotely shiny or a large granule that is considered like calcified material, not exactly a microorganism. And when we see those little things over there with some form of compartment inside and some membrane inside, those are considered eukaryotes or just other microorganisms, fungi of the sort. Now we have the garlic mustard, the white snakeroot, and the Japanese knotweed. Now I cannot exactly deduce how much diversity there is within each slide, because that involves a lot more testing and does not involve microscope evaluation. It involves genetic testing, and I couldn't do that. But I just wanted to touch upon the fact that just how beautiful it is. You know, when you look at it, it's just fascinating to see that there's so many little things inside the soil that are just mediating everything and determining, oh, the nutrition will go here, there, and they just have their own system within that is assisting both plants and each other around. In the garlic mustard, you can see there's a lot of evidence of microorganisms in the area. Now, if it's other forms of bacteria or eukaryotes, that I do not know, but it is evident that there is budding going on, so those are microorganisms. As for the white snake root, there is a lot of bacteria and some granules. A lot that I found that might be just because of the small sample size, but otherwise that is evident in that sample. As for the Japanese knotweed, we see that, well, that has a high concentration of organic matter, but that little guy we see in there, I deduced, is a microorganism. Now, it's a rotifer or some other microorganism of the sort that thrives in the soil, that I do not know, but it is a microorganism. And as you can see, that's those as well, or so my observations. As I stated, there's a greater nitrate concentration in the invasive samples for the nitrate quick test. The control yielded greater value nitrogen concentration as opposed to the two native samples. And the lower concentration calculated from native species that were in the vicinity of invasive species was done. And that is evident in both what we had deduced in the previous slides. As for the microscopic evaluation, now I can't exactly say how much the diversity was or at what the biomass exactly was, but there was a greater observable abundance of microorganisms in both the Japanese knotweed and the garlic mustard rather than the white snake root. Now, again, that might be due to sample size, but we will see. So my main conclusion is that invasive species are actively restructuring the microenvironment in the soil to further their advances in propagating across the area, in which they deplete all native species of their nutrients and affect the microbial diversity from within the soil. They are now greatly affected from the changes that were done to the microbiome and that were originally there to facilitate order and maintain good health. So this brings me to my final aspect of my presentation, which I find is critical to understanding how we can utilize these microorganisms that mediate the soil to see other ways to eradicate invasive species. And that involves genetically engineered microbes, also referred to as other parts as biocontrollers. So as for microbes as being biocontrollers, there are a lot of companies. I think there is one referred to as New Leaf Biotech. They are currently responsible for creating microorganisms that are engineered for crop culturing and for eating the soil and the nutrition from within to sustain crop and to let them grow to their fullest potential each season. And their research regarding microbes and their versatility in eradicating destructive invasive plants still continues to rise. However, I do want to preface this by saying that this area is heavily under-researched, and I could only find several reviews detailing how microorganisms could be used to invasive species, but the fact that there isn't enough funding or enough dedicated research being put into it means that there isn't a lot of applications being used for these. But there are a lot of examples, for example, with other invasive species that aren't confined to just plants. That includes mosquitoes, because we know Disneyland, they're doing a lot with eradicating female mosquitoes, and the way they do that is they genetically engineer the mosquitoes to then only have male mosquitoes reproduce and the female mosquitoes die off. So, and as I said, other examples of this already exist, but that can be expanded upon with maintaining or altering the microbiome within genetically to then eradicate invasive species. As for how, microorganisms can act as pathogens, and we know this by having infections within us all the time, and the same with plants. However, there is evidence of this happening naturally in wildlife, so therefore, why can't we access it now to eradicate invasive species? Different types of bacteria engineered to target specific aspects of the invasive plant, so they could specifically target root growth or other chemical releasing mechanisms within the roots of each plant, and that can be crucial for eradicating invasive species as well. And they can also supplement native species or beneficial exotic plants because microorganisms are responsible for doing just that. And if the microorganisms are being harnessed by these invasive plants chemically, then by stopping the chemical flow from the invasive plants, they can actually allow the native plants to reproduce effectively and grow. So other further potential recommendations on this is to advocate and fund for more research, use measures to preserve microbial diversity in the soil and target invasive species specifically, and to remember that microbes facilitate the balance in order needed to preserve the natural environment. So this is kind of a avant-garde way of approaching things in terms of eradicating invasive species, but it is crucial to find other paths to eliminate them and to preserve the natural environment. Limit the use of pesticides and herbicides in the long term and be more effective in supporting native plant growth, and so as to not spend a lot of money in terms of physically taking out or using other measures that are detrimental to the natural environment and spending, I think the US spent over 400 million in terms of eradicating invasive species from 2014 till now. And that is a large number. So hopefully by using this method, it could lower the price. So I would just like to now acknowledge the Verona Environment Commission for providing me access to do this presentation, for helping me with research, for allowing me and Cece to sit in and watch the meetings and go by and understanding how exactly the commission works to address these topics. So I'd like to thank the Hilltop Conservancy for providing me information on the statistics of how many invasive plants are in the area and just providing me an area to which I can test. Verona educators as well. Without them, I don't think I would be able to learn any of this or have the means of actually presenting this. I would also like to thank Dr. Siglishvili and Dr. Lunsford. The way I did my tests and the microbial analysis is by going to their lab and having access to their equipment as well. So that's the end of my presentation. Thank you.
Nia, when you get questions, repeat them and then answer them.
Repeat the questions.
When you get a question, repeat the question so that it's on the mic.
All right. So
Sure, I have a two-part question, so it's gonna be difficult if you repeat it. So are invasive species always a threat when introduced to new environments? I know you talked about depleting nutrients, and if that's the extent of it, maybe you could confirm that. Otherwise, maybe you could just talk a little bit more about it. So that's the first part of the question. The second part of the question is, do the three invasive species that you focused on in your research pose a threat to your environment, to our environment? If so, could you explain the nature of the threat? And I'm sorry if you already addressed that. If so, I'm hoping you could maybe explain it in terms that people without the benefit of your scientific background could understand.
okay, so the first question was, actually, could you repeat it again? Sorry.
These are always bad.
Okay, so whether or not invasive species are always bad when introduced in the environment. So originally, we could say that invasive species aren't going to take an immediate effect on the ecosystem as a whole. However, when introducing invasive plants, they can have qualities that are not as similar to the surrounding plants in the area. So given that they originate from a completely different continent or a different area or habitat as a result, just the different qualities that they have, the chemicals that they release into the soil, it could be detrimental to other plants as they have not been exposed to that originally. So as a result, they can populate at greater speeds, they can take in more nutrients as a result because they have qualities that aren't as similar to the surrounding plants, which they haven't been exposed to, if that makes sense. They're almost like a virus in the grand scheme of things. So
And the second question was about the nature of the three invasive species.
So there was two invasive species. One of them was the garlic mustard and the Japanese knotweed, and asking like how to what extent their damage could put onto the environment.
What kind of threat it poses?
So the garlic mustard I have read upon is really it's a great example for a plant that really takes the nitrogen out of the soil and really just infuses it to themselves and doesn't let any other plant have it. So it's a really good example with respect to that, because without the necessary nutrients for the native plants themselves, they are able to take that those nutrients and assist themselves to grow. And they do this by, as I said, like chemical interactions within the soil. They can alter the microbial diversity from within, and when they alter the diversity, that means that the microorganisms are now going to supplement them, and they can also eradicate the necessary microorganisms that are respective to the native plants. And as a result, the native plant will die off because it doesn't have the necessary nutrients it needs from the microbes in it. And the invasive plant, the garlic mustard, will survive. And the same goes with the Japanese knotweed. It has a very similar trend in terms of taking those nutrients and extracting them and putting them into themselves, rather than the native plants, which do not have that quality. Yes.
Okay, so my question is an invasive species, can it be harmful to other invasive species?
That's actually a really good question. So, in terms of, so the question was that how could invasive species be harmful to other invasive species? And honestly, yeah, because just like any other plant, or every other animal in the world, they want to survive. So when an invasive plant, which is bad for native plants, and also another invasive plant, which is also bad, comes together, well, they're not gonna be friendly with one another. They're going to affect each other both in the ground and on top. They're gonna fight for the soil that they want. So, which is why you can see some invasive plants, they might die off and they won't survive, and others will thrive because it's the type of environment that will allow them to live like that. So, oh yes.
I using it's
Mhm.
so the two that you talked about, the mustard one and Japanese one, which one do you think, if they were fighting for space, which one do you think would win?
That's a really good question. Definitely the knotweed. And actually, I do have physical evidence of this, because when I went onto the Lenape Trail, or from where I collected the samples, all I saw was the Japanese knotweed. It was taking over, and it's very obvious that the Japanese knotweed is just the winner because it just takes over all the time, and the garlic mustard it can't fight. It can't fight.
And depends on what. Why they're taking on.
Why they're taking?
Yeah.
Well, they take over because of how fast they grow. So also the speed of how they grow. And that is also determined by the microorganisms from within. Because the more nutrients they have, the greater probability that they'll grow and they'll keep propagating and just scour and just cover every single piece of land in its vicinity. So that is exactly it. There's also I forgot to mention, there are foragers, and foragers being other animals that eat the plants themselves, which can also allow invasive plants to just continue to grow. And that's not exactly a microorganism issue, but that is also another aspect that allows invasive plants to grow.
First, I want to say, after the first presentation,
I thought to myself, I would not want to go second, but you guys were both equally. So I thought I knew a little bit about invasive species. I've never heard of garlic mustard.
Mhm.
I like garlic. I love mustard. I never think of anything as that. My question is, during your presentation, you mentioned that sometimes removal can contaminate even at a greater level.
Mhm.
Do you recommend removal or containment? Or does it depend on the situation?
That's a good question. So the question was whether the removal will affect, or will removing the invasive species be better or worse than rather containing them, and if I'm interpreting that right. And honestly, it depends. Because right now, the best course of action is also removal and using other types of chemicals that are either synthetically or organically created to eradicate these species. But as of right now, I think we just don't know a lot about invasive species because each one is different in their own respect, each species. So how you eradicate or eliminate them, it's really difficult to pinpoint. So whether or not we can keep physically removing, I think that is the best course of action. It's just a matter of understanding how far we will progress to maybe not having that be the best course of action anymore. So, yes.
Okay, so. I just like that.
That's okay. That's okay. Yes.
So Nate, were any of the three nitrogen fixers to start with? Your
Um,
garlic mustard, your
The garlic mustard was, yes. The garlic mustard and the Japanese knotweed. Wait.
nitrogen fixation.
Yes.
Did all three of them undergo nitrogen fixation or just the two invasives?
All three, I think.
Okay,
Yeah.
as you know what I'm talking about, the nitrogen nodules, they all had, okay, is that why you picked those three or because they were all together?
So, I mean, they all go through nitrogen fixation. I picked specifically the garlic mustard and the Japanese knotweed because from previous research review, and I think there was one done in Virginia, they detailed how the garlic mustard and the Japanese knotweed are really popular in the sense that they fixate nitrogen at a faster rate than the white snake root or other native plants in the area based on the data that they collected. And also they had the diversity index for the garlic mustard and the Japanese knotweed was really high in comparison to the native plants in the area. So, which can be connected as a result to the increased rate of nitrogen fixation. So, yes. Yes.
Okay, I kind of have a clear question. So first, have you done other research about different plants?
If I've done other research on different plants, I think I've gotten super focused on those plants that they stuck in my head the most. So I remember only from that. Yeah.
Okay, my second one. Have you and Cece done anything together? Like, have you worked on like any species together?
So if me and Cece both work in the same areas,
Yeah, like if she was next to a river, if she was in a river and then you were like right next to her, have you ever done like that kind of thing?
we haven't done that. But I do want to say, me and Cece's projects do go hand in hand because they're very similar in that respect. Because water quality also affects soil quality and vice versa. So that, in a way, that's a connection. Yes.
I find it fascinating that you use the garlic mustard and the knotweed, which are both edible plants. And the snake root is a poisonous plant. In fact, Abraham Lincoln's mother died from poisoning of her milk from her cow eating the snake root. Now, you also found out that the bacteria level in the snake root was lower than the other two. I'm wondering if now you piqued my interest is the snake root actually killing off the bacteria to give it a better way of surviving in that soil and now allowing other plants because the snake was that white flower thing you see, and that you must have found a lot of that where you were located and was blocking out all the other native crops and taking over and killing off with the poison that is in the because it's an alkaloid in the snake. Did you notice that, or did any other plants that were living around the snake root being dying off?
I mean, I didn't see a lot of, well, I did see a lot of white snakeroot as the summer ended and kind of
Yeah.
the fall came out. So, I mean, I actually don't know because in terms of how diverse the microorganisms are within, now I can't exactly deduce, but I can say that whether the nature of the plant is poisonous or not can affect the surrounding organisms as well. But it also comes down to just how within the plant's rhizosphere itself, just how the microorganisms are interacting with one another. And I feel like the common trend is that the more diverse the microorganisms, the better the plant grows. And as for the white snakeroot, it wasn't holding up a lot in comparison to the Japanese knotweed and the garlic mustard in the area. So I think it's like almost the opposite, and where the white snakeroot isn't populating as much, and it's just not affecting the surrounding
Right,
invasive
so you're
plants.
saying that the nitrogen in the knotweed and the other was actually accelerating the growth, which then would overtake
Yeah.
the environment.
Mhm.
So that's your right.
Any other questions? Yeah. Sound wonderful again, just amazing. Thank you. Take a picture if you want to move it and say thanks to.
Yeah, yes.
That was fine. So,
Yeah.
yeah, Stan, is that okay? I just want to get reviewed at that.
All right. Thank you, everybody, for coming. I really appreciate it.
That is so good.
And thanks to these two students. Shut up, Sarah.
I just let them